Okay, good evening, everyone. So let's continue quickly. So every one of you very quickly answer my question. What is the name of the enzyme that is involved in removing three phosphates? What is the name of the enzyme that is involved in the removal of the three phosphates? Every one of you, what is the name of the enzyme that is involved in the removal of the three phosphates? Every one of you, what's the name of the enzyme that is involved in the removal of the three phosphate groups? I hope every one of you remember, right? We generally call it as phosphohydrolase enzyme, or you also call it as what enzyme, friends? You also call it as the triphosphatase enzyme. Okay, please don't get confused and don't forget why people are not answering. Okay, every one of you very quickly answer my question. Uh, from the see, you remember, right? From the mRNA we remove one phosphate, and if you everyone quickly recall, even from the GTP also we will remove two phosphates. My question to all of you is, from the GTP which phosphates we are removing, friends? My question is, from the GTP which phosphates are removing? Hope every one of you remember, right? Very good. From the GTP, friends, we are removing the beta phosphate also, as well as the gamma phosphate also. Whereas, if you look into the mRNA, friends, in the mRNA, we have removed only alpha phosphate. Correct, na? I believe every one of you remember. Last and final question. Who is the donor of the methyl group? Who is the donor of the methyl group? Who is the donor of the methyl group? Very good. So the donor of the methyl group is generally called as what, friends? Adomet, which we generally called as adenosylmethionine. Please don't get confused with that particular point. So friends, till here we have discussed in the yesterday class, that is called as 5' capping. Hope every student remember, after the capping, what we have to do, friends? We have to do the process of the polyadenylation. Yeah, so polyadenylation. I have told you very clearly, friends, since we are using the word called as a polyadenylation, how many number of the adenine molecules we have to add, friends? We have to add nearly 250 molecule, 250 adenines we will add. More number of adenines we are generally adding, friends. Now all the students listen my good evening, continue. All the students listen my statements. If you quickly recall, I have explained it to all of you the major functions of all this particular what we can say that 5' capping. And similarly, friends, I want you to understand what is the major function of the polyatyl. All the students keep this point in your mind, friends. The ultimate goal of the polyatyl is, listen to me very carefully, which enhance the lifetime, and I can tell you very clearly, friends, which is required for the translation. Translation, even it is required for stability. So remember, uh, 3' polyatyl is the one which increase the lifetime or lifespan, increase the stability of the mRNA, and most importantly, friends, which is also involved in the process of translation. We will discuss how in the translation. And just now we all people studied that, friends, in case of a prokaryotic organism, we all people studied there is only involvement of the 3 enzymes. But friends, if you look into the eukaryotic organism, all the students remember there is a involvement of 6 proteins. Please do not get confused with that particular point. Okay, all of you try to understand my statements without fail. No, don't get confused. Listen to me. You remember where we add the polyadenylation on the mRNA, friends, there is a specific site is located. What do we call that particular site, friends? We generally call it as the poly A site. So friends, what we are doing actually, we will cut that particular poly A site and we will add it. Is it or not? Of course, yes. So how many number of polyadenyl we will add? Nearly 250 adenines we are going to add. And friends, the most important point that all the students has to remember here, we will cut at the poly A site. Now the question is that, friends, whatever the site that we cut, what happens to that site? That's very important, right? Friends, that particular site will undergo the process of degradation completely. Now listen, no need to get confused. Let me explain to all of you how does the process of polyadenylation will be taking place. Every one of you pay attention and look into the diagram very correctly. Friends, this is mRNA. Remember, every one of you remember, at the 5' end of the mRNA, at the 5' end of the mRNA, there is a specific sequence is present. AAU, AAA. And in the 3' end of the mRNA, there is a sequence called as GU rich sequence. And you can see this, friends, this is called as a poly A site, where we are going to cut that particular site. Every student commonly know about that. Now the question is crystal clear. Does every single mRNA contain the same kind of a sequences? Friends, I am using the word general. I'm using the word general. So I can say that not all mRNAs. Friends, please listen my statement. I am not using the word all mRNAs. I am using the word most of the mRNAs. Most of the mRNAs having this particular AAU, AAA sequence and GU sequence at the 3' direction. Please don't get confused with that particular point. Now it's very important, friends. You people has to remember the name of the enzymes which are involved here. What are the name of the enzymes? CPSF. I encourage everyone to remember the short names only. Cleavage and polyadenylation specificity factor CPSF, cleavage stimulatory factor CSTF, cleavage factor 1 CF1, cleavage factor 2 CF2, and PAP poly A polymerase, and PABP2 poly A binding protein 2. See, friends, you commonly know that what is the function of a poly A polymerase. This is the one which is actually adding more number of the adenines. You know it very well. Now what is the function of this particular PABP2, friends? The point is that poly A polymerase cannot add adenines continuously. Hardly it can add 20 to 30 adenines only. That's all, friends. Now the question is very clear. If we want to add more number of adenines, the poly A polymerase compulsory require the help of this particular protein. That is what we call it as PABP2 protein. Please don't get confused with that particular point. Okay, now all of you listen. I'll explain in a simple words. Listen to me correctly. So what we have to do, friends, we have to cut. Where we have to cut? We have to exactly cut at this particular poly A site. Every one of you. After cleaving that particular poly A site, here only, friends, we are going to add this particular poly A. Now you can see, right? CPSF, the pink color one is the CPSF, blue color one is the CSTF protein, and the green color ones are the CF1 and CF2 proteins. Now all the students look into the diagram very carefully without having a confusion. CPSF, the pink color protein, will attach to the mRNA exactly where? Exactly at the AAU, AAA sequences. You got the point? And friends, where exactly the CSTF will be attached? The blue color CSTF exactly attaches at the GU rich region. Now you know what happens. Whenever, listen my words, whenever the CSTF attaches, all the students listen, CSTF will bend. Listen to me carefully, friends. CSTF will bend the mRNA in such a way that it will bend towards the poly A region. You can see that. It will bend towards the poly A region. Then what's going to happen? Immediately the cleavage factor 1 and cleavage factor 2 comes into the picture. And what is going to happen? They will cut at that particular area. The moment that it cut that particular area, now this entire area will be removed out. It will be degraded automatically. Now the enzyme like a poly A polymerase enzyme will come. But what I told you, poly A polymerase, they will do only the process of slow polyadenylation, very slow. So hardly it can add, friends, 20 to 30. That's all. Immediately, friends, whenever what we can say that PABP2, when the poly A polymerase associated with the PABP2 friends, remember all the students, rapid polyadenylation will be taking place. Please don't get confused with that particular point. This is what we can call the polyadenylation, a very simplified mechanism. Then in the post-transcriptional processes, if you get a very difficult question, there is a possibility that you will get the very difficult question from the splicing. Now, you know very well, the simple words you use it to remember. Splicing means removal of the introns and joining of the exons. Everyone use it to say that splicing. But friends, trust me, splicing is not that much easy step. Splicing is a very complicated step. I want every one of you to understand that correctly. Why it is a very complicated step? All the students listen. This is the exon, you know very well, which is called coding part. You also know very well this is the intron, non-coding part. Why I am using the word splicing is a very accurate process is, suppose friends, even by mistake, remember every student my words, even by mistake, even a little amount of the exon got cut. Remember my words, even by mistake, a little amount of the exon got cut. What happened? The coding information is getting lost, which leads to the very dangerous mutations. That's why, friends, you can say that undoubtedly DNA replication is the step which is the most accurate step. After the DNA replication, friends, I can say that the splicing is the most accurate step. I want you people to understand that very correctly. So what is splicing? Formation of a mature functional mRNA. So what we are going to do here, we are removing the introns and we are joining the exons. Please don't get confused with that particular point. So now look into this. Now this is the mRNA, right? This is the mRNA. Look into this picture very correctly. Now the question is here, friends, same. The sequences whichever I am showing in the diagram, will it be present in every single mRNA? No. Again, I can tell to all the students, friends, it is again general sequence only. It is again generalized sequence, which means that which is present in most of the mRNAs. All the students listen. This is called 5' exon. This is called intron. All the students listen my statements very carefully. The entire process of splicing, listen to me. The entire process of the splicing is exclusively depends on the branch point. Exclusively. And there will be another exon. So listen my words very clearly, friends. Where we cut the intron, that's the question. We never cut the exon, okay? We never cut the exon. We always cut the intron. Where exactly we cut the intron? Every student listen. We cut the intron exactly. Every one of you pay attention. Exactly at the 5' splices, at the splice site. Now, friends, the question is that why did I call it as the 5' splice site? Because the splice site is located towards the 5' direction. Now, here also, it is called 3' splice site, friends, because it is located towards the 3' direction. Such a simple statement it is. Now, very important point all the students has to remember, at the 5' splice site, there is a sequence called GU. And at the 3' splice site, there is a sequence called AG. Now, does this sequence is common in every mRNA? No. Most mRNAs contain this particular sequence, but it is not common in all. In your exam, they can ask a direct question in the exam. Which of the following mechanism can be followed in the process of splicing? Every student remember my words. Friends, the splicing mechanism can be shown transesterification. Never ever get confused with that particular point. What is that? Transesterification. Removal of the intron and joining of the exon. Let's see how it is happening. All the students remember, again and again I am telling to all of you, the entire process of the splicing is exclusively depends on the branch point. There's no doubt about it. Now look into this correctly. Sorry. This is the branch point. See this is the 5' exon, which is located towards the 5' direction of the mRNA. Everyone pay attention. And this is the 3' exon, which is located at the 3' direction of the mRNA. So this is what, friends, this is the branch point. Every one of you know very well. Now, all the students, listen my statements very correctly without having any kind of a confusion. Just follow me. You know, right, a very common point, wherever the splice site is present, there what group is present? Listen, at the splice site, friends, there is a presence of the phosphate group. Please don't get confused with that particular point. Now, all the students, listen my statements very correctly. Now, the branch point contain what group? Branch point contain OH group. This is only more important step. Follow me. Branch point contain OH group, where the oxygen can act as a nucleophile. What is nucleophile? Oxygen having a lone pair of electrons. It will do the nucleophilic attack. What is nucleophilic attack means? See this. This oxygen will come and cut the phosphate group. Which phosphate group, friends? The phosphate group present at the 5' exon. As a result, what's happening, friends? Half of the intron came out. You can see this. This entire intron came out. Now, friends, you can see this picture, right? This intron came out. But still, friends, we have a little more intron is left out on the 3' direction. Now, what is happening now? The OH group present at the 5' splice site, friends, it will go and cut. Listen, everyone. It will go and cut this particular splice site, which is present at the 3' direction. Don't get confused with that particular point. As a result, what is happening, friends? The entire introns will be removed out and the 2 exons will be joined. Every one of you quickly answer my question. Here, which enzyme will come and join the 2 exons? Every one of you. Everyone, which enzyme will come and join these 2 exons? Everyone, quickly. What's the name of the enzyme? Undoubtedly, ligase will come and seal this particular nick. You all people commonly know about that. And if you look into this picture, friends, very, very carefully try to understand me. Whenever the intron comes out, how does the intron look like? Everyone listen. Whenever the intron comes out, friends, it look like a structure called as lariat. I already told you, friends, there is no scientific reason behind this. Why do we call lariat, friends? I told you now. Farmers used to hold the neck of the cow by using the rope in this way. So they will make the rope according to the structure of the neck and they will hold it. So whenever the intron comes out, friends, intron also will come out in the form of this shape. That's the reason only we are calling it as the lariat intron. No need to get confused with that particular point. Okay, now all the students listen. Remember, friends, there may be a different type of splicings that you people can observe. But what is the most common statement that every student has to remember? It doesn't matter how many number of splicings are present. The only matter is what? In every type of splicing, mechanism is same. Types of splicings are different, but mechanism is same. That is the transesterification. Now let's try to understand the different types of splicing. So friends, look into this. There is first one called as self-splicing mechanism. Then next one is called, friends, listen everyone, spliceosomal mediated splicing. And third one we can call, friends, alternative splicing. We will discuss one by one. It's not required actually, but I'll explain till the spliceosomal mediated splicing. That's more than sufficient for you. Alternate splicing, but remember the types of splicing. Trans splicing. So there are total 4 different type of splicing mechanisms are there. Now you listen to me very correctly. In all the 4 different type of splicings, what's the common point? The common point is that mechanism is the transesterification mechanism. That is a common point. Now the question is that how does the self-splicing introns or differ from other mechanism? See, friends, name itself indicating, right, self-splicing, which means that it is not, listen everyone, it is not at all taking the help of anyone. So with the help of a branch point, the introns will cut automatically. We don't use any external enzyme, we don't use any external protein. that is the reason only we called it as the self-splicing introns. Please don't get confused with that particular point. Now, all the students, listen, friends. What are the different types here? Group 1 intron, group 2 intron. I will tell you the difference. Before the difference, you have to understand what is common. Common is that group 1 and group 2 never ever depends on anyone. Just with the help of branch point, the splicing occur. But what is the difference between the group 1 and group 2? Just listen. Group 1 is found majorly in the nuclear, mitochondrial, and chloroplast genes, which are coding for rRNA, mRNA, and tRNA. In the afternoon class, we discussed, right? Friends, mitochondria and chloroplast, they can code for their own tRNA, then own rRNA. So in there, I don't know whether any one of you remember, in the afternoon class only, I have shown you one thing. Friends, the chloroplast is actually, I shown you in the blue color. The chloroplast particularly contain an intron region. You remember? Anyone remember? That intron is what? Group 1 intron only. The next, group 2 introns. Where the group 2 introns are found, they are found in the primary transcript of same mitochondrial or chloroplast mRNA of exclusively fungi, algae, and plants. You have to remember it compulsory. Yes. Now, listen, I will tell you, friends, the difference between the group 1 and group 2. What is the common? The common point is that they will show the mechanism of self-splicing, which means that there is a no protein or there is a no enzymes are involved. That is called self-splicing. So then, friends, what about the group 1 and group 2 introns difference? Listen, look into the point. See, friends, the only difference, the mechanism of the group 1 introns is they are having a branch point. Listen, everyone, they are having a branch point. But the point is that, friends, branch point do not have the internal OH group. That is the only point you have to remember. What is that? In case of a group 1 intron, there is a presence of the branch point. But the point is that the branch point do not have internal OH group. So which means that, friends, someone has to donate this OH group. Who is going to donate that OH group? That is the question. Guanine. Every student remember, guanine can act as an external donor of the OH group. So you look into this picture, friends, everyone pay attention towards the diagram. This is the 5' exon, this is the 3' exon, and this will be the branch point. What I told you just now, I told you that the branch point do not have the OH group. So friends, it is going to recruit the OH group externally. Now the question is very clear. Who is the donor? Who is the donor of that particular OH group? So who will donate? Guanine. This is the only point you have to remember. Guanine donate the OH group. The moment guanine donate the OH group, every one of you look into this. So oxygen acting as a nucleophile, you know very well. Now, friends, the oxygen will come and, remember everyone, will come and cut this. You can see, right? Will come and cut the 5' splice site. So, friends, now you see this, half of the intron came out. Now, then this is called first transesterification mechanism. And during the second transesterification, you only try to understand, again, this OH group will go and cut the phosphate group present at the 3' splice site. Then, friends, the intron will be removed out and the 2 exons will be joined by the ligase. But the interesting point here, you can see this. In the intron, again, there is a OH group is present, and that OH group will go and cut the phosphate group present within the intron itself. As a result, some intron come out, remaining intron undergo the cyclization. You don't need to remember that. What I want you to understand for the exam is, in case of a group 1 intron, remember my words, guanine is the one which is acting as an external donor. That guanine, which is having the OH group, that will come and cut the phosphate group present at the 5' splice site. As a result, half of the intron came out. And remaining, what I want you to understand, that OH group will go and cut the splice site present at the 3' direction. Then the entire exon come out, and most important point, friends, the introns will be joined with, exons will be joined and the intron will be removed. But what is the point? Here the intron is not lariat shaped completely. You can see, right? We can call the cyclized intron. And friends, if you look into the group 2 intron, same mechanism. This is the same common mechanism shown by the group 2. Why? Because friends, group 2 having their own internal adenine. It is having, where is the middle point? Where is the middle point? Where is the middle point, Anusha? Okay, now listen. What is the specialty of a group 2 intron? They do not have any, they are already having their own branch point and their branch point having their own OH group. So friends, group 2 introns do not require any external guanine to form the lariate intermediate. Group 2 having their own adenine. So what is the specialty of the group 1? What I have written? Group 1 only do not have, they are having the branch point, but the branch point do not have the OH group. What's the point? You have to remember, the OH group is coming externally from the guanine. So the difference is this. This is the difference. What is the common similarity? Did we use any enzyme here? Did we use any protein here in order to cut the intron? No, we did not use any external protein. We did not use any external enzyme. So what's the point you have to remember? We have shown the mechanism called as self-splicing. Never ever get confused with that particular point. Okay, now listen to me very correctly. So we understand the first type of splicing, friends, we can call it self-splicing. Now you listen to me very, very correctly. Okay, now spliceosomal introns, we also call it as what, friends? Large introns. Now the question is very clear. Just now, friends, we have studied about the self-splicing introns. Correct now? What is the specialty of a self-splicing introns? They will do the splicing on their own. They don't require any external enzyme. They don't require any external protein. That is called self-splicing. Now, friends, name itself indicating, right? Spliceosomal introns. What do you mean by that? Listen, everyone. If this particular intron wants to be removed, remember my words correctly. If this particular intron wants to be removed, there is a compulsory requirement of a protein complex. What is the name of that particular protein complex, friends? We call it as the spliceosomal mediated complex. Please don't get confused with that particular point. Okay, now all the students try to understand what exactly I'm trying to explain to all of you. Just listen, just follow me very correctly. Remember my words correctly. Spliceosome, it is made up of two things. What was that? Spliceosomes are made up of RNA molecule, and friends, the spliceosomes are made up of the proteins also. There is no need to get confused with that. It's made up of RNA and it's made up of the proteins also. Every one of you listen my statements correctly. To listen, RNA and proteins together, we call it as SNRP. What is the full form of a SNRP? Small nuclear ribonucleoproteins. What's the full form? Small nuclear ribonucleoproteins. That is called SNRPs. Only RNA, it is called SNRs. Small nuclear ribonuclear RNA. See, everyone listen, no need to confuse. You have to understand this correctly. The spliceosome is made up of a specialized RNA protein complex. We call it as the SNRP complex. Each SNRP contain what? Each SNRP contain, listen every one of you, it contain RNA. You know what is the length of that RNA? 100 to 200 nucleotide. This is called RNA. Now, friends, in this 100 to 200 nucleotides, which will be present more, more uracil. So this is called U1. U1 means many uracils are present. U2, again uracils are present. There is a no U3. Why? Because in this particular area, no uracil. Then followed by U4. Then followed by U5. Then followed by U6. Like this, there are many uracil regions are present. I will explain to all of you once again. Follow me. So what does this spliceosome is made up of? Spliceosome is made up of RNA plus protein. RNA plus protein together, we are calling it as the SNRPs. Only RNA, we call it as the SNRs. So one RNA is having the length of 100 to 200 nucleotides, and it is having abundant amount of the uracil regions, which are designated as U1, U2, U4, U5, U6. Now look into the question very correctly. Now the point is that why do we use this spliceosomal complex? What's the reason? What is the reason we are using the spliceosomal complex? The reason why we are using this spliceosomal complex is that all the students try to understand my statements very correctly. We are using because the intron, whatever we want to remove, it is a very large intron. So the intron can't be removed on their own. They are not self-splicing intron. They are very large intron. That's why, friends, we use the mechanism called as a spliceosomal intron mechanism. Friends, please don't get confused with that particular point. Okay, you got that very correctly. Now, all the students listen. How is this mechanism is happening? Look into this. Simple point it is, look into this. This is 5' exon. You can see that. And this is the 3' exon. You can see that. You can observe that at the 5' direction, there is a splice site. At the 3' also, friends, there is a splice site. You got the point. And you can also observe, friends, there is a branch point. You can see that very, very clearly. Now, what's the point you have to remember? The point is that branch point cannot do splicing on their own because the introns are very long. So branch point compulsory require the help of other proteins. Now let me explain in a simple way. Every one of you pay attention. Who is coming first? Every one of you listen my statements. Who is coming first? First, U1 will exactly come and attaches to the 5' splice site. And friends, where exactly U2 will come and attaches to the branch point, near the branch point. Now the question is that, friends, how does U2 comes to know it has to attaches exactly at the branch point? How does U1 comes to know it has to attaches to only the 5' splice site? Friends, they are all having complementary sequences. Every student, look into this picture very correctly. Look into this, every one of you. This is the mRNA, right? This is what, friends, 5' splice site. And what was this? This is the U1. So U1 having exactly, look into this, the complementary sequences near the 5' splice site. That's why U1 attaches there. And if you look into the U2, U2 exactly attaches near the branch point. Why? Because you can see, right, U2 having the complementary sequences near the branch point. Please don't get confused with that particular point. Now look into this. Look into this. So what's happening now? U1 attaches to the branch point. U1 attaches to the 5' splice site. U2 attaches to the branch point. Then you know, friends, what's happening? U1 and U2 going to recognize. The question is crystal clear. What the U1 and U2 going to recognize? That's the question. U1 and U2 going to recognize another 3 complexes, U4, U6, U5. We can call it ternary complex or tertiary complex, widely called ternary. Now U4, U6, U5 will come and attach us here. Now you see this, what are all present? U1 is present, U4 present, U2 present, U6 present, U5 present. Now you know, friends, what happens when all attached with each other? All the students remember my words. Structural changes are happening. What changes? Structural changes are happening, or conformational changes are happening. Look into the point. Because of the structural or conformational changes, look into this. U1 is getting released out. At the same time, friends, U4 is also getting deleted out. I mean to say dissociated. U1 dissociated and U4 also dissociated. Now all the students look into the picture very correctly. Now what are left out? Now friends, the left out is U2, U5, and what is left out, friends? Another one is what? U6. These 3 are left out. Now you only look into the picture. What is our ultimate goal? Our ultimate goal is transesterification. Look into the picture. Look into the picture. The U2 attaches where? You can see, right? U2 attaches exactly near the branch point. You can see, right? U2 attaches exactly near the branch point. Now, so what's happening now? This particular U2 will push the branch point towards the 5' splice side. See the arrow. U2 will push the branch point towards the 5' splice side. Now the branch point which is having OH group, it will cut this splice side. Then what's happening? See this? Half intron came out. Now you see this now. Now this OH group will do the second transesterification, which means it will go and cut the phosphate group present at the 3' splice side. You got the point very clearly, right? Then what's happening? Entire lariat intron will be released. U2, U5, U6 will be released out, and the friends exons become spliced with each other. There is no need to get confused with that particular point. So once again, all the students try to understand. Let me quickly explain to all of you. Why do we use this spliceosomal mediated complex, friends? What is the reason? Because branch point alone cannot remove the intron, so it is taking the help of spliceosomal complex. So in the spliceosomal complex, U1, U2, U4, U6, U5, variety of uracil rich regions are there. You can look into the picture. First, U1 attaches to the 5' splice site, and friends, U2 attaches to the branch point. Then what's happening? U1, U2 recruit U4, U6, U5. Then all come close together. As a result, structural change or conformational change occur. As a result, U4 and U1 will be released out. And left is U2, U5, U6. So what's going to happen now? Look into the point very correctly. The U2 will push the branch point. Look into that. It will push the branch point. So branch point having OH group, right? Now that OH group will come and cut the 5' splice side. So friends, half the intron came out. Then what's going to happen now? Now this particular OH group will do the second transesterification. Cut the phosphate group present at the 3' splice site. Then the complete intron comes out in the form of a lariat. Two exons will be joined by the enzyme ligase. U2, U5, U6 will be released out. So since we have used the spliceosome, that's why this splicing is called as spliceosomal mediated splicing. No need to get confused with that particular point. Right. Perfect. Okay. Is it clear, every one of you, about the splicing? Let me know quickly. Everyone, if you have any kind of a doubt, let me know right now. Clear for everyone? Quickly. Anyone having any doubt, I will re-explain. Come on, come on. You should be very, very active in the last movements. Okay. We should never give up until the end. Okay. 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 That's fine. That's fine. Okay. Someone having doubt. Yes, in correct, Shravanti. In the self-splicing only, we are taking the help of the external OH group, which is donated by the guanine. Right. Okay. No. Shravanti, we are not using any enzyme. You are getting my point. Just guanine is not an enzyme, no? Guanine is not a protein, no? Just guanine is a base which is donating the OH group, that's all. When we can called self-splicing is when we don't use any external enzyme. When we don't use any external protein for the splicing, then you can call self-splicing. When you use external enzyme and external protein, then you don't call it as self-splicing. You got the point? That's great. Okay. Okay. Now we are entering into the last step of the central dogma. What was that? That is called translation. Every student very commonly know that the ultimate goal of our DNA molecule is synthesis of a protein. Correct? Now, there's no doubt about it. Now, the point is here, you also know very well, DNA is a highly stable molecule. In order to protect its integrity, in order to protect its stability, the DNA molecule is going to synthesize RNA instead of synthesizing the protein directly. Now, you know very well, whatever the protein that I want to synthesize, Where that information is present? That information is present in the DNA. Suppose I require this region of the DNA, which is used for the protein synthesis. So this region converted into RNA. Now the RNA is going to give rise to protein finally. So now listen how many names we can give. DNA don't involve in the protein synthesis directly, but RNA does. So what I can say? I can say, friends, RNA mediated protein synthesis. See the word I am using. RNA mediated protein synthesis. That is called translation. I can also say another word, DNA guided. DNA guided, RNA mediated protein synthesis we can call translation, because friends, DNA is everything. Just imagine DNA is like a hard disk in the computer. Without hard disk, system never work. So what's the point? You have to understand DNA, friends, it will store the complete information. So DNA will guide it, RNA will follow it, finally we synthesize the protein. You know very well, in our body or in a plant body, in a bacterial body, in any organism, if any organism want to function properly, they require protein. Without protein, not even a single cell also work properly. Of course, Balakrishna, we can say that RNA dependent protein. We don't use the word dependent actually. Okay, we don't use the word dependent. We can use the word RNA mediated. Why don't we use the word dependent? Is because RNA is not the one who carries the information. Correct? Now, DNA is the one who carries the information. DNA is giving the information to the RNA. Please do the synthesis of a protein. So RNA is not dependent. RNA is guided. RNA is mediating the synthesis of a protein. That's a simple point here. Now, so very important, whether it is a prokaryotic organism or the eukaryotic organism, the process of translation undoubtedly will take place in the cytoplasm only. There's no doubt about that particular point. Now, the very important point you have to remember, whether it is a prokaryotic organism or the eukaryotic organism, whenever the protein got synthesized, the protein never or the protein never acquire biological activity. That's a very important point, friends. The protein is a biologically inactive protein, immature protein. So inactive protein, to make it mature, we have to do again some modifications. What do we call it, that modifications? Post translational modification. Again, where they are happening? Again in the cytoplasm. So now my point is, whether it is a prokaryotic organism or the eukaryotic organism, the process of translation and the process of post-translation, both are exclusively happening only in the cytoplasm. Never ever get confused with that particular point. It's easy to remember and easy to understand. Now look into that. Now, if you ask me a question, which process is very difficult process? Undoubtedly, I can say compared to the replication and transcription, translation is a very difficult process. The reason is that there is an involvement of multiple enzymes. There is an involvement of a multiple factors, and the time taking is also too much. So that's why, see this. Yeah, the time taken for the translation is more and more complex. So now see, Balakrishna, what we can say. Translation is what? mRNA directed. It's not a dependent. mRNA directed biosynthesis of polypeptide. Now, what's the interesting point? You all people very commonly know that in the nature, there are more than 300 amino acids are present. But what's the interesting point? Out of 300 amino acids present in the nature, we use only 20 amino acid in order to synthesize the protein, because the human gene, now I don't use the word human gene, I'm generally saying that most of the proteins, I don't use the word most of the protein, I can say all proteins can be coded by 20 different amino acids only. Now, you also know very well, if the process of translation wants to occur, you require tRNA, you require ribosomes, and most importantly, you require many number of translation factors. We require, see, you remember, right? In the transcription, what we studied, we called transcription factors. In the translation, we can call translation factors. Many factors required for initiation, elongation, termination. Every step there is a requirement of many number of processes are required. No need to get confused with that particular point. Now look into that. Now friends, if we want to synthesize protein, information is coming from where? DRNA. Who is bringing the information? From the mRNA. How it will bring the information? Look into this. Everyone know very well, you studied in the 12th standard also. This is the DNA, correct na? DNA contain what? Nucleotides. So you also know very well, there are 3 bases on the DNA. There are 3 bases on the DNA come close together. They called it as a codon. When 3 bases come close together, then you can also call it as a triplet. So most of the students won't think here. Some student, you all people has to ask me one question. Sir, why only 3 are coming together? Why can't 2? Why can't 4? Why only 3 are coming? Why do you call triplet? Why don't you call doublet, or why don't you call the quadruplet? Because friends, only when the 3 bases come close together, 3 bases only can code for an amino acid. See, the reason is very simple, friends. This is the tRNA, right? tRNA contain what? Anticodon. Every student know very well, the anticodon contains how many bases? Here also 3 bases. Here in the mRNA, it contain only 2 bases. How it can code with the tRNA? So always remember, that's why in the DNA, 3 bases come close together, they can form the codon. So friends, whatever the information that is present in the DNA, the same information will move to the mRNA. Correct na? Now you always remember. See, I don't want you people to focus on how we read this particular genetic code. What is the method of reading? You don't get in the exam, but I'm just telling you as an overview. Always see friends, genetic code never ever be read in a random way. You should always read in a sequential manner. And very important, you should not read the codons in a reverse orientation. That is completely wrong. While we are started reading the codons, friends, we should always start reading from the 5' direction only. Never ever get confused with that particular point. So listen here, 5' UAU code for tyrosine. So CUA code for leucine, so on. So 3 bases come close together, they can form a one specific amino acid. This is the sequential way of reading. I don't want you to focus on this reading. I want you to just read a PPT slide, because we don't get that in the exam. Just for your understanding, but I want you to have a look of that. How can we read it? Now, which is very important, listen everyone, based on the reading frame, all the students listen, based on the reading frame, there were total 64 codons were identified. Based on what? Based on the reading frame. Now, you know, right, in many number of exams, SET also you got, in the JL also you got, in the DL also you got. In your upcoming exams also, there is a possibility that they can ask you, ask the question in the exam. You know very well. Anyway, let me ask you a simple question. I hope you all people remember. Give me the name of at least 2 amino acids. Give me the name of at least 2 amino acids which can be coded by more than 6 codons. Give me the name of the amino acid at least coded by more than 2 codons. Sorry, more than 6 codons I'm talking. Sorry. Everyone, you forgot. If you are not answering, trust me, which means you are not reading. How many number of times I told you to read and remember the 64 codons. Do not neglect it. Look into that. Leucine, serine, arginine. Serine, arginine, and leucine. These three are the amino acid which is coded by more than are coded by 6 codons. So now friends, you all people very commonly know that out of 64 codons, 3 codons cannot code for any information, which means cannot code for any amino acid. So that's why we can call stop codon. UAA, UAG, UGA. We generally call it as the stop codons. Don't get confused with that particular point. Look into that. Now, friends, you know, right? AUG. My simple question to all the students, very simple question. Do you think the AUG can code for the methionine in all organisms? My question is very clear. Do you think AUG can code for the methionine in all the organisms? No. See, now, but can I tell you one statement? You tell me, can I say AUG is present in every organism? Can I say this? Can I say this? AUG is present in every organism. Of course, yes. But I cannot say that AUG is going to code for methionine in all the organisms. Remember, very good. Even GUG also code for methionine. Now, that is not my point. Suppose in certain organism, AUG code for something else. Suppose you look into the picture. Every one of you look into the picture. UGA, you know very well. Just now we seen here. UGA is ultimately is going to code for the stop codon. But what's the interesting point here? Now the UGA is not going to code for the stop codon. See, remember, in certain organisms, it is coding for tryptophan. In Drosophila, in tryptophan, even in some plants, it is coding for tryptophan instead of coding for the stop codon. AUA originally has to code for the isoleucine, but in some organisms it is coding for methionine. So friends, see, that's the reason. What is the statement I have written here? I have written a statement that they can give the same statement in the exam. The standard genetic code is widespread. What is the meaning of widespread? It is present in every organism. Every single organism, the genetic code is present. But what's the point? It is not universal, which means that it is not going to code for the same information in every organism. Present in every organism, but it is not going to code for the same information. That's the point. So AUG code for one codon. Listen, you have to remember, friends, there's no shortcut here. And leucine, serine, arginine. These three are the amino acid which are coded by the 6 codon. And threonine coded by the 4 codon, alanine, valine, glycine, proline. They are also coded by the 4 codons. So if they give the question in the exam, what will you do if you don't remember them? It's very important to by heart all the 64 codons. You can't interpret from where you can where you can expect the question. It's very idealized to read all. Okay, now listen. Just now we all people discussed very clearly that if the process of the protein synthesis wants to occur, we require tRNA, we required mRNA, and we require rRNA compulsory. So without tRNA, mRNA, rRNA, it is very difficult. Protein synthesis cannot occur. Every student commonly know that tRNA, mRNA can be synthesized in the nucleoplasm, rRNA is going to synthesize in the nucleolus. Now, what's the point? The point is that now whichever synthesized in the nucleoplasm, nucleolus, now all are coming to the cytoplasm. Why? Because all has to be assembled in the cytoplasm. Then only the protein synthesis will occur. And I also told to all of you last time also, friends, when compared to the all type of RNAs, tRNA is the type of the RNA which is having a less lifespan. Never ever get confused with that particular point. Always remember that point. Now look into that. Look into that. So what I'm trying to explain to all of you. tRNA, once it gets synthesized, it got synthesized successfully. The moment it got synthesized, it undergo foldings. It undergoes, I don't use the word modification, I'm using the word foldings. It will undergo the foldings during the time of modification. During the time of modification, it undergoing the foldings and it look like a clover leaf-like structure. Whenever it undergone the foldings or the modifications, it can give rise to loop and stem structures. What? loop and stem structures they are going to give rise to, they are going to give during the time of modification. You can call D loop, you can call T loop, you can call the anti-codon loop. Very, very important. Similarly, you can call the D stem, T stem, anti-codon stem. Very important all are. And very, 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 very important for the exam point of view is to which direction or to which point of the tRNA the amino acid will come and attaches. Tell me quickly. To which point or to which direction of the tRNA the amino acid will come and attaches. Every one of you. Undoubtedly towards the 3' direction. Every one of you remember that point. And at the 3' direction, what sequence is present, friends? There is a presence of a ACC sequence. This is also most common sequence that is present in the tRNA. Please don't get confused with that particular point. Now the question is here. So listen, all the students listen. Everyone try to understand me what I am saying. Amino acid will come and attaches to the 3' direction. I understand it very clearly. Now the question is, friends, who is guiding? That's a good question. Who is guiding the 3' hydroxyl group to recognize the amino acid? Someone has to guide it. So who will guide? D loop will guide. So D loop will guide the 3' hydroxyl group of the tRNA to recognize the amino acid. Now this is the mRNA. mRNA having codons. From where the codons are coming from? DNA. Now to this codon, the anti-codon has to be associated. This is called anti-codon loop of the tRNA. Now again the question is that who is guiding the anti-codon loop to recognize the codons present on the mRNA? That's an important question. So who is guiding, friends? Undoubtedly the T loop is guiding. So what's my conclusion? T loop is guiding the anti-codon loop to recognize the codon present on the mRNA. D loop is guiding the 3' hydroxyl group of the tRNA to recognize the amino acid. That's how the tRNA will be ready. Right? But still it is not activated. We will discuss in the tomorrow class. Is it clear, everyone? Till here? Please revise it once again without fail. Perfect, perfect, perfect. Okay. Take some rest and once again start doing the revision. Don't sleep more time. Just sleep 5 to 6 hours enough. Okay. Try to spend more amount of the time on your self-study. Okay. Perfect, perfect. Okay. Thank you. I'll see you in the next class. Okay. Thank you.